to a new video for YouTube from Rubex Biology. This video that we have today is again a Quora questions video. This is going to take you through an important question that we have been asked, or in general, it's prevalent over at Quora, and so we'll take you through it, explain the concept behind it, and I've actually picked a pretty interesting one for today's class. So the question that I have picked is: HIV is a retrovirus. How are retroviruses unique from other viruses? So this is what we are talking about today. Firstly, we could just begin with a brief description of what retroviruses are. Retroviruses are mostly, let's just do this. Yeah, they are mostly ssRNA viruses. And how do they replicate? They replicate via a DNA intermediate. The important enzyme which is involved in this process, we have to remember its name. It's RNA dependent DNA polymerase. So this is the important enzyme which is involved in this process. What happens here is that RNA is used as a template, and what is produced from it? DNA transcript, or maybe a DNA copy is produced from it. Since there is conversion of RNA into DNA, this is the reason why this is known as reverse of transcription. Right? So this is what happens here. Let's just discuss the rest of the process too. DNA copy duplicates and forms dsDNA, that is a double-stranded DNA. What happens with this dsDNA is that dsDNA integrates into the host genome and creates many ssRNA viral progeny. So basically, there is replication with the help of a DNA intermediate. Let's just discuss this. So we have a simple ssRNA virus now, right? This ssRNA virus is going to serve as a template. So I can just mention template using the text box. तो आपका template जो है वो बन जाता है ssRNA. What happens is that it forms a complementary sequence of a DNA. Right? So what is formed is a DNA. I'll just show it in a slightly darker way to distinguish it from the RNA. So let's just do it like this. DNA को थोड़ा अलग दिखा दें हम. Right? So it forms a DNA transcript. After that, one copy of DNA forms its complementary copy too by simple duplication. So basically, the ssDNA now gets changed into dsDNA. दूसरा कॉपी भी यहाँ पे DNA का ही बन जाता है. So what is being formed now is a dsDNA. This dsDNA goes inside and becomes a part of the host genome. तो ये host genome के अंदर जाके, if you can see, it gets fixed inside your host genome. After that, it creates many single-stranded copies of progeny virus. What kind of progeny? Just like the parent. Progeny, ssRNA virus. So what is happening here? It's reverse transcription. Why? Because RNA is getting changed into DNA with the enzyme that we just discussed, RNA dependent DNA polymerase. This enzyme is also known as reverse transcriptase. That's the name of this enzyme. If I have to show to you this thing with the help of an image, okay, here it is. If you just go through it, explaining the process simply using this, my pencil. So if you just see here, you have this ssRNA template, and an RNA DNA hybrid is being formed. They have shown DNA in orange color. From the single-stranded DNA, they form a dsDNA by simple duplication. Now this double-stranded DNA integrates into the viral genome. It becomes a part of the host genome, So it becomes a part of the host genome, and then it creates many single-stranded viral RNA progeny. So it helps in formation of the progeny of the virus then. This is the enzyme that I told you, also known as reverse transcriptase. So I hope our Quora question was clear to you all. And for any more of you, any more doubts that you all have, please let us know in the comments, and we'll be coming up with videos for them quickly. So by the time, just like, share and subscribe our video, and stay tuned for more upcoming ones. And thank you for whatever encouragement you've shown us in the past, and I hope it continues. See you all. Stay tuned for the next video. Bye. Don't forget to like and subscribe.